Michele Kosiewicz, PhD. Why are men more protected from lupus, and can the reason lead to new therapies? Thank you so very much for joining us.
Okay, so I'm going to talk. I'm from the University of Louisville. I am a PhD scientist, and I am an immunologist. And I'm going to talk to you a little bit about hormones. As you all know, I'm sure, lupus is women are at much higher risk of developing lupus than men are. So there's a really strong gender bias there, 9 to 1. 90% of all patients who have lupus are women. So the question is, why are women at much greater risk? Or the alternative question, which is what we're interested in, it's a little bit opposite, because we think we can utilize that information to maybe help patients with lupus. Why are men at much lower risk of developing lupus? So this is the question that we addressed. I'm not going to talk a lot about the preliminary information I was going to talk to you about, because Dr. Krager already did that. Fortunately, he already laid the groundwork, so when the hook comes out, I hopefully will have finished. But as you know, that there are genetic bases as well as immune regulation and environment. Dr. Krager had mentioned that already, that impact and determine whether or not you develop lupus. So just a little bit of a primer here. Sex steroids are involved in immune response. We know that the female sex steroids, the estrogens, including estradiol, do exacerbate disease. They're generally immunoenhancive. That means that they increase the immune response. They don't decrease the immune response. And many cells in the immune system have receptors for estrogens. So that means that the impact can be on many different cells in the immune system. Androgens, which are the male sex steroids, and testosterone is one of those. And again, there are receptors on all immune cells in the immune system that can be responsive to androgens, and they generally are immunosuppressive. So the simplest answer to this is that the sex steroids that women have, estrogens, are immunoenhancive, and so you're actually creating too much of an immune response, whereas men have androgens that are immunosuppressive, so they suppress immune response. It's not that simple. If it was, life would be much easier to deal with. So just again, in general, the estrogens or the sex steroids, pregnancy can exacerbate, as some of you may know, increase complications and flares. Exposure to estrogens, either in the form of oral contraceptives, hormone replacement, even fertilizer, insecticides, estrogenic compounds you can find in the environment, can all potentially exacerbate disease, theoretically. Androgens, we know that androgen levels correlate inversely with disease activity, so women do produce some levels of androgens. And if you see a flare, when you see a flare, generally the androgens are lower, so that's called an inverse relationship. Female and male lupus patients tend to have lower levels than androgens than non-lupus patients, and we know that people with Klinefelters, those are men with Klinefelters, have an XX. Again, they have lower levels of androgens and a higher susceptibility to higher incidence of lupus. And chances are patients probably do metabolize androgens differently. And we know in mouse models, and forgive me because I'm going to talk about mice today, so, and you're probably going to be happy that this isn't being done in humans yet, so just bear with me. We know that if you castrate males, so if you remove the source of testosterone in models of lupus, that males develop disease. So they don't even have to have estrogens. It's just good enough to be able to eliminate those androgens and they get disease. And if you treat females with testosterone, not something that I would recommend for females because the side effects are not good. Very, 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 very strong, very negative side effects. You can actually prevent disease in female mice. And this is just our model, our animal model of lupus, an exceptionally good model for lupus, except that they are mice. And this is just an age. So they do get autoantibodies, double-stranded DNA antibodies, all the same antibodies that human patients get. And this is a mouse model of glomerulonephritis, so nephritis, so kidney disease, and they do get kidney disease. So in blue, you have the males, they don't get disease. The females are the only ones that get disease in this model. And you can see survival. So the females, in this model anyway, have survival issues. And this is just information telling you that in this model, when you castrate males, they start developing autoantibodies just like females. And if you treat females with testosterone, they don't get disease. So again, this is a very complex disease. That's why it's been so difficult to develop drugs or any kind of treatment for lupus. So I'm going to focus on, because my lab works on immunoregulation, how the immune response is regulated, and the interaction with the environment, which we consider hormones to be part of the environment, in a sense. And I'm going to talk to you about, these are the happy cells. Dr. Krager had Tregs, and next to it he had a happy face. So these are cells that suppress the immune system. They control the immune system. They're a special type of white blood cell, leukocyte, that regulates or suppresses other cells when necessary. Okay, so in people who have different autoimmune diseases have defects in these cells. So these cells are really important. If we can get these cells to be functional and to get more of them, that would be a really good thing, and we can control autoimmune disease, but it's very difficult to do that. These cells are made in several locations, and they can be actually made by other kinds of immune cells, and I'm telling you that for a reason. Now, your eyes are probably going to glaze over when I start talking to you about the data, but look at the top of each of these slides, and it's actually the take-home message, okay? So, for example here, these are Tregs, and they're about, in our animal model, females have about 25, sometimes as much as 50% fewer of these regulatory cells than male mice do. And these are a special kind of regulatory cell that are induced in so they're made by those other immune cells I was telling you about. So females do not make regulatory cells in this model as well as males do. And that's going to be an important issue. Okay, I won't go into the GI tract because we're going to talk about the GI tract a little bit and why that's important for this system that I'm telling you about. Dr. Krager already told you about all the different parts. So this is, if you take the colon or the intestine, stretch it out like a hose, and cut it, the inside of that hose is where pre-poo is formed, basically. It's where the bacteria live, and it's where you digest the food, okay? So that is, that is here. So this is the internal. This is a cross-section. This is the cell layer that divides, that separates you from everything that's, a little bit too much caffeine. Okay, so, and these are the cells that we're interested in. They're actually embedded in the wall of the intestine on the inside, not the outside where the bacteria are. And these are cells whose job it is to make regulatory T cells, these Tregs. That's one of the jobs that they have. That's these guys right here. Okay, these are the cells that we're interested in. And in this experiment, what we did is we asked how well those cells make those regulatory cells that we want to make. And we compared them between males and females. And we found, and my graduate student always tells me, tell everybody it took her 2 years to do this experiment, okay? So it's a very long experiment and very energy intensive. These female mice just don't do it as well as males do, okay? They just don't make the regulatory cells as well. And this is another experiment that confirms that. If we castrate males, if we remove the androgens from the males so that they don't produce testosterone, they look very similar. So even though these dots don't look the same, these are bars, they do not produce, so if you castrate males, they do not produce regulatory cells. So they look a lot like a female, basically. So we know that androgens are important in making these Tregs, in making these cells. Again, don't let your eyes glaze over here. These are gene expression assays. And so what we wanted to know is what was the difference between the female and the male cells that made those female cells not very good at making T regulatory cells. And we found a gene that was defective in those females. Okay, so just keep in mind the phrase retinoic acid. Okay, remember that. That's a molecule that's made from vitamin A, okay, really important molecule. If these cells don't produce that molecule, they cannot induce regulatory T cells, okay, so remember that. Okay, so just a summary and conclusions of this part. In general, normal numbers and function of regulatory T cells, or Tregs, are required to prevent and to control autoimmune disease, okay? We know that that's true for all autoimmune diseases, including lupus. Females and castrated males that have, they have low or no androgen are at higher risk of developing lupus. We know that that's true. Female and castrated males have lower numbers of Tregs. Okay, so that kind of makes sense, right? The immune cells that help make those T regulatory cells are defective in both female as well as castrated males, suggesting that it's the androgens that are probably controlling the function of those cells, okay? And we think that this defect is due to inadequate levels of retinoic acid being produced. Again, remember that, retinoic acid. So strategies that can increase the ability of those cells to make regulatory T cells or produce retinoic acid might be very effective therapies for the prevention or the treatment of lupus, okay? You with me so far? Ish, right? Okay. This is just a pretty picture. So now we're going to talk about microbiota. So I'm that other investigator that he mentioned. It's like, okay, so what does that have to do with anything we're talking about? So all of those cells that we said are involved in making Tregs are in the gut. So they're going to be exposed to the bacteria that are in your gut. So, and the gut, and I'm not going to go over this, you have trillions of these little bugs in your gut. I mean, trillions of them. And they're good bugs by and large, as long as you don't have some sort of odd disease. And there are thousands of different kinds, but there are trillions of these guys. They way outweigh the number of cells you have in your body. You just have lots of them. So they have a huge impact on many, many functions in the body. And this is just this complicated picture here, all the different things that the microbiota in your gut do, okay? They're involved in producing vitamins. They break down non-digestible fiber, metabolize complex carbohydrates, they synthesize they do all kinds of things. What we're interested in is they are also required for the normal development of the immune system and immunoregulation. Okay, if you don't have normal microbiota, you're not going to develop a normal immune system, and that can create problems. So that's the tie-in. Oops. Okay, this is just a pretty picture that one of our graduate students painted, and this is of the gut that I can't resist. So this is the gut, these are the bacteria, this is the inside of the gut, this is the bacterial wall that we showed about. So these are the cells that we're interested in. These are the ones, I know they kind of look like creepy crawly things, right? But these are the cells that actually induce the regulatory T cells, okay? And we think that these have an impact on autoimmunity. Oops. So, the gut microbiota and disease. Now we know the gut microbiota has a huge impact on diseases that are at different places in the body, not just in the gut. Response to viruses, issues with the nervous system, development of immune system, I mentioned already, hepatic disease, type 1 diabetes, obesity, inflammatory bowel disease, of course, and we think autoimmune arthritis, and we think that it also has an impact on lupus. Okay, there was a recent paper, and this is what made us actually go after these kinds of, do these kinds of experiments, so to speak. So apparently there's a relationship between gender, between sex, between the gut microbiota and autoimmune disease. And this was originally discovered in type 1 diabetes, but we wanted to see if there was a relationship in lupus, because in type 1 diabetes there is no gender difference like there is in lupus. So we asked the question, does gender affect the types of microorganisms in the gut of lupus-prone mice? Okay, and the answer is yes. So male mice have a significantly different composition of microbiota than female mice do. We don't know if this is true in humans. We'd love to do the experiment. We'd love to do those studies. Those are, in the future, these are all preliminary studies. So it is definitely the case in adult mice. Males have definitely a different composition. Okay, so the next question is, okay, different composition. What happens if we take basically the feces, it's the microbiota from males, and give it to females? Transfer that microbiota in. And so what we find, so we give it to in what I would call a mouse that's at the tween age, okay? Peripubert, around the time of puberty, so we start feeding them at that time. And we give it to them monthly. And we do feed the feces. Basically, we call it a poo shake. And it is cecum, and it contains microbiota. It contains the microbiota, okay? It contains the actual bacteria from the males. If we feed female cecum, cecal contents microbiota, you can see that they start developing autoantibodies. This is the normal trajectory, the normal time course of this in this model. However, if we feed male feces to these females, we can actually significantly suppress disease. This model is very, very difficult to treat. It's very difficult to prevent disease in this model. You have to give them massive doses of steroids. This is a remarkable prevention. I mean, it really is. And this is the nephritis, so kidney disease is dramatically suppressed in these animals as well. And survival is really dramatically suppressed, increased, sorry. Mortality is suppressed, survival is definitely increased. This is just by feeding microbiota. So the question then is, if we give the reverse, so that's if we give male feces and give it to females. What happens when we give female feces and give it to males? Does that mean they get disease? And the answer is no, they don't get disease. So if you give the female microbiota, so in this model system in lupus, it does not help to treat with antibiotics, okay? Antibiotics do not do anything for lupus. It does not prevent lupus in females. It does not help lupus at all in this system, okay? So in this system, the male microbiota are protective. It's not the microbiota itself that are causing disease, but the males are producing something that makes them be protected from lupus, okay? So if we then, remember those cells I was telling you about that are involved in inducing T regulatory cells? If we feed the feces and then look at those cells in females, I know it's hard to tell, but just take it from me, those cells are now able to induce regulatory T cells, make regulatory T cells in response to being fed the male flora, okay? And again, the Tregs go up, okay? By feeding male flora, male microbiota into the females, we can actually make the regulatory cells go up. Okay, so then the question is, what about fecal transplantation? It probably doesn't sound too appealing, but it's a possibility. We now know that fecal transplants are being given to different kinds of patients for different diseases. People who have antibiotic-resistant Clostridium difficile infection, who are very, very sick and resistant to any kind of treatment, have literally been saved by being given a transplantation. But that's an entirely different situation. That's an intestinal infection. There are clinical trials for ulcerative colitis and Crohn's disease, which are form of inflammatory bowel diseases, and irritable bowel syndrome. Those are in clinical trials. We have no idea what the potential is for the treatment of autoimmune disease in humans. We just don't know that. There are cautions that we have to think about if you do transplants. Whose stool do you use, right? What does it contain, right? What does it have? What does it contain? So one person's stool might be okay for one person, but not for another. So we just don't know enough about that. There's too much in the stool. There's too many different things that can happen that we just don't know how that can help. So it could exacerbate symptoms or cause other pathologies or complications. So this is a possibility in the future, but it's very complicated and it's very difficult to do that. So what we did is we asked, okay, so if we don't know what the bugs are, which we have an NIH R01 to look at the actual microbiota itself, but for the ALR, for the Alliance for Lupus Research, the funding there is we want to look at the function of the microbiota. So what are the microbiota producing? What are they producing? Because that'd be a lot easier if they're producing something they can just feed rather than having to figure out what to give them. The microbiota produce lots of different things, okay? These are all the different categories of molecules that the microbiota produce in general. Uh-oh, am I about to get dragged off? But this is sorry, I didn't even see my sign. Sorry about that. So this is just the take-home message there, and this is just a schematic of that. We think this is probably not the only metabolite, but our ongoing research, we're treating right now with phytol, okay, and we have some preliminary results that are promising. We have another analysis coming up where we're looking for other metabolites that we might be able to create a cocktail for treatment, possibly. And we also want to identify the mechanisms and the cells that are involved. And these are the people. Most of this work was done by Anita Shabra, a very talented graduate student in the lab. And thank you very much, and sorry for going over time.